This morning, because I have two sections of this class, before my morning class, I sort of got this itch, and I started looking online for different pictures that depicted different habitats as we went through geological time on Earth, and I was getting really fascinated. We're kind of getting into it. So I found this one picture I wanted to start a class with today, and I'm actually going to kind of gear class today as sort of a tour of the scenery while we talk about the plants. And I kind of wanted your opinion on this picture. Where do you think you would be on Earth that you would see a habitat like that? Quite obviously not in Florida, right? For one thing, it's got them big hills in the background and things called mountains. We don't have any of that. But where do you think you would? Where do you think you need to go on Earth to find something, a place similar to this, with this kind of vegetation, this kind of? Yeah, me too. That's what I thought too. I was thinking Ireland, Scotland, New Zealand. I agree. Any of those places that we normally associate with being, like you guys said, moist, not a lot of sun, not what we'd say an extremely harsh habitat. One that kind of is conducive to these low-growing plants, these mosses, right? So, when you're looking at that and you're starting to think about where we are in terms of talking about the evolution of plants, this picture that you're looking at, even though it looks like a pretty good picture, it looks almost like a photograph. If you look a little more closely, and probably some of you are better than I am, if for no other reason than your eyes are better, that's not a photograph. It's a computer-generated picture. And what this is an image of what we think it looked like on Earth about 450 million years ago, which is where we are in the time scale right now talking about plants, okay? Last time we were together, what we finished up was we talked about the mosses. We still have to go over the actual mosses today, but we were talking about the non-vascular, the seedless plants, those low-growing. Osmosis is okay, diffusion is okay. I'm small enough, I can survive that way. Again, and it isn't a matter, like I've said before, of these organisms being better or worse, because there are still mosses around today. So in some habitats, in some areas, that particular lifestyle still works very well, right? They're not a dominant species, right? They're not everywhere, but nonetheless, there are several different species of mosses, right? So if you think about these areas that we mentioned, Ireland and New Zealand, these moist, sort of mild type of climates, rocky types of habitats, that's kind of what the Earth was like 450 million years ago. And if we go to those places today, we find that what does better in those kinds of places are the kinds of critters that we think 450 million years ago began doing that first, right? So it all makes sense. So we can borrow evidence from the present time based on the kinds of conditions we think occurred back in the past and say, okay, well, the organisms that live in those kinds of environments today probably are very similar to the organisms that lived in those kinds of environments back then, because it's all about the environment, right? It's all about the environment. It's all about adapting to the environment, and it's all laws of physics. You're gonna have to stay low to the ground. You have to be a small little plant if you're not vascular, right? If you have vasculars, if you have vessels, you can grow larger because you can move food and move nutrients around. But if you're small, you're kind of limited to diffusion. All right, you're sort of limited to diffusion and osmosis. So and that works, but it doesn't work if you get too big, right? Diffusion and osmosis can't penetrate really deep layers of cells. It can only go so far in. So let me find this other picture that I was going to show you, because I want to show you what the Earth looked like as a whole. Is it that one right there? Yeah. So here we are coming out of the Cambrian. All right. Notice where the continents are. Located down around the southern end. Still north is north and south is south, just like it is today. The only difference is, they haven't tipped this, obviously, to make it indicate the tilting of the Earth's axis. But this is where the continents were, right? And that's why sometimes it's funny, or not funny, but it's very interesting when we find like tropical species in the fossil record down in Antarctica. Remember I told you Antarctica is a continent under ice, but when we get down into the rocks sometimes, they'll find remnants of these tropical species. We'll talk about, we already did lungfish, right? And they go, oh, that's interesting, And people say, well, maybe Antarctica wasn't cold at one time. Well, it wasn't, but it wasn't because the South Pole was cold. It's because Antarctica, it wasn't at the South Pole. It was more like at the equator. All right, so notice what's happening though during this Cambrian period. We have these continents, we have these land masses that are forming. Jared, yes, remember you have your exam on Tuesday. You also have lab, and then on Thursday we'll meet online. Okay. All right, so we've got these land masses that are forming, and the interesting thing about when we're forming these land masses is anytime you start forming land masses, you start forming diverse, different habitats. Hi, Rayanne, how are you doing? Sorry, I didn't see you come in. Jared, I don't think I said hello to you as well. I'm sorry. And so when we get all these land masses forming, what we get is variability. We get variability of ecosystems. We get variability of environments. We get, like, for example, near, let me get a different color. Blue on blue is not going to work very well. Blue, let's make it white. There we go. So near shoreline areas is going to be one type of habitat. You remember I said in the ocean, I think I mentioned to you guys, it might have been my marine science class, I don't recall exactly, but I said there's not a lot going on out in the middle of the ocean when it comes to productivity. There are phytoplankton and organisms that eat those, but most of the productivity is going to be near the shoreline. That's because, for one thing, it's shallow enough that light can penetrate down, and you can get bigger plants for more photosynthesis. And a lot of that has to do with a lot of the nutrients that are necessary for the plants to grow actually are runoff from the land, like when the rain falls down and leaches certain chemicals, like calcium, for example. Marine organisms need a lot of calcium, especially the ones that make shells. Those mollusks that we'll get into, that's what their shells are made of. Yeah, they're made of calcium. All right. So what we're getting in as the late Cambrian, so this Cambrian explosion time, and just a little bit before the time we're talking about here, 50 million or so years, is we're getting a lot of variability of habitats. And of course, along the coastline, you're probably going to get tropical, wetter habitats. That's the same as today, right? Islands will have unique habitats. And then as you get further inland, and this is going to become a lot more prominent about 200 million years in the Permian, is you get areas inside the continent begin to dry up. Okay, because they're further away from the ocean. They're further away from a big source of water vapor. So we're going to have dry habitats, wetter habitats, and everything in between, right? It doesn't go from desert to tropical rainforest across the street. It doesn't work that way, right? So anytime, and of course I've driven it into your head by now, anytime we have differences in the environment, we're going to have organisms that are diversifying to take advantage of those different environments, right? Now, many of them are going to try and fail, but some of them are going to make it. Quite a few of them make it, obviously, because there's quite a few of these organisms around today. All right, so this is what the habitat, or what the Earth looks like. Let's look at the habitat going back a little bit from that first picture I showed you, before we have this Ireland-looking habitat. There it is right there. Kind of want to show you the progression. So here's the Ordovician period. Now the Ordovician period is right before the period that we're getting ready to talk about today. So this is the period in which the low-dwelling mosses, holy cow, I just drew a blank. The bryophytes, the organisms that we were talking about, the hornworts, the liverworts, there we go. This is the kind of habitat they might find themselves in. And notice even in this picture, again, this artist rendition, but notice that even in this picture, we got diversity of habitat. We got those that are these little squiggly things that are living near the water. We got a little bit drier climate. And as you get further away, and it's greenish in color, so it looks like something's making a living there, you got drier environments, okay? So this forming of land, this forming of the continents that occurred in the evolution of the geological evolution on Earth was incredibly significant, right? If for no other reason, well, for many, many reasons, that it gave many thousands and upon thousands and millions of opportunities for organisms to adapt adaptations. If the environment always stayed exactly the same, right? You know the answer to that. Nothing would change. Wouldn't have to change. It's only when the environment changes that you get speciation. All right? And sometimes that environment changes slowly, relatively slowly. We might get gradual changes. Remember gradualism? Sometimes that environment might change a little more quickly. Or sometimes it's not so much that the environment changes quickly that would contribute to, say, like punctuated equilibrium. It could be that organisms, as they move out through these different environments, they encounter a whole bunch of different environments, kind of like the Galapagos finches did, folks. Galapagos finches went through a punctuated equilibrium, adaptive radiation, many species evolving, not because the environment changed a lot, but because they found themselves, they have migrated, moved out into different environments. So as you get these different variety of environments, we might have had a lot more of those punctuated equilibrium events, okay? So here is, this is the mosses, the low growers. The environment is still not horribly conducive for living out too far away from land. There might be a few in the background that are eking out a living, sort of the, like I was saying about the mosses, the green scum types, the algae, and then of course moving off from the algae, the green algae, we move in, like we said, the mosses and those organisms, right? Okay, so after the Ordovician, we get the Silurian. Let me show you what the Silurian looked like. I love these pictures. You get online and just it kind of takes you there. That's what I love about these this ability to generate. Look at this. Now this is significantly different, right? So during the Silurian period, we have this diversity of habitat, but quite clearly what's going on, looks like in the background, is they're trying to show you that maybe the volcanoes are starting to mellow out a little bit. So we're not getting such noxic fumes in the air. Maybe a little more water is collecting. So we're getting a moister world, and certainly we're getting diverse habitats as well. You can see up here that certainly looks a little bit drier. But along these areas where there's water, and we know that life has started in water, we got these plants that are starting to stick their heads up, being a little bit larger than those plants that you find growing low to the ground. These are the organisms here that can live, a little more than just the mosses when it comes to this whole, living so close to the ground. This is a plant we'll talk about in just a second, actually. This is Cooksonia. Cooksonia is a plant no longer around, 425 or so million years ago. It's very prominent. The little yellow things you see at the top, those are the sporangia. Those are where produce the spores. There's actually plants that are alive today. It's very weird. There's these things called living fossils. Living fossils are things that are alive today that look like things that existed millions of years ago, or that we thought had gone extinct, and then we found one that was alive. Like, for example, in terms of these plants here, Cooksonia is extinct. It's not around today. But I'll show you a little bit later on something called a whisk fern. And whisk ferns look an awful lot like Cooksonia. So, and they're not related. They're not related at all. They're very distantly related, but they're not related in terms of close relationship. So this is, I'm sorry my voice is changing. I'm getting up to open the door. But this is kind of like convergent evolution in a macro scale, right, over millions and millions and millions of years. And I'll show you the plant in just a second. And that plant that exists today, those whisk ferns, only grow in tropical or subtropical climates. And to me, this kind of looks, this looks pretty doggone tropical or subtropical. Certainly looks a lot wetter and moister than what we might find in some of the drier climates. Now, it doesn't mean those drier climates weren't around. Okay, it doesn't mean that we didn't have those dry environments, but we also had a lot of environments in which there was really abundant plant growth, especially along the edges. Let me pull up now the Devonian. So we're moving up into the next era. There's the Devonian. And what happened in the Devonian, especially in the later part of the Devonian, and again, this is about the time that starting just about when we were talking about the mosses, moving into the end of the mosses. Again, it looks like the volcanic activity may have died down even further, right? And the sky's kind of brightening up. So we're getting a lot more sun. Sun. Plants, if you got sun, one of the things you can do as a plant to give yourself an advantage is grow tall, especially if you can grow taller than your neighbors. But it looks like we got these, it looks like a desert kind of scene, doesn't it? So, during this time, we have, again, this diversity of habitats. It doesn't mean that all the tropical habitats were gone, but it means that organisms, again, evolution is driven by the environment changing or organisms moving into new environments. So these organisms are expanding out, and they're finding out ways to survive in these drier climates. Now, soon after the Devonian, or right after the Devonian, comes a period that is named for what happened during this period. And that was the picture that I first showed you. Oh God, where is it? Is that not the picture I first showed you? I'm sorry. Where did I put that picture? Well, anyway, the next period that came about, I must have deleted accidentally, is a period of time we call the Carboniferous. And the Carboniferous is named that because if you dig down through the rock layers, you remember they named these geological periods and eras based on what the rock layers show us. But if you go down to the Carboniferous about 375 million years ago, so kind of right after the mosses that we were talking about the other day, you get this really thick layer of carbon, very thick black layer of rock. Okay, and of course coal, right? That's what coal is. And coal is the organic matter of living organisms. And back, if you dig down into the Carboniferous era in the rock layer, it's a really thick layer. So we know that there was massive forest growing back during that time. Okay, so we see a dry climate here, but we see other areas on Earth soon after this as the continents were moving around and climates were changing, and some climates were becoming wetter and some were becoming drier. We see a rapid diversification of plant growth causing this Carboniferous period. And at about this time, this is what the planets look like. This is when Pangea comes into existence. All right, that big supernet. And again, just think about all the diversity. You got all of this shoreline. You got even little rivers. You got little inlets right here. You probably got mountain ranges. You got dry areas that are inland. You got tropical areas that are near the coast. And then you got everything in between, right? I mean, you just got a smorgasbord of environments for organisms to live in. Now, were these organisms, all organisms, able to invade all environments? No, of course not. No, we've said that already, that what we're looking at when we look at the organisms that are alive today are the ones, the lineages on the tree that made it. All right, but if you're trying, and there's lots of organisms trying, then there's a better chance that some of you are going to make it. Okay, so now let's go back for just a second and take a look into our lecture slides and go back into the mosses. I didn't finish up. We did talk about the liverworts and the hornworts. But now let me load the slides. Thank you. There we go. Right. So what I said, we're about 450 million years ago. That's where we are. That's that picture that I showed you at the beginning. Picture that looked like Ireland. So that's what it looks like. So 450 million years ago, we got this environment that's very conducive for plant growth. And then as we went through the rest of theirs, we noticed that what was happening is we got many different environments. And that's going to stimulate, obviously it's going to stimulate evolution. So we talked about the liverworts, the hornworts. Let's talk about the true mosses today, and then we'll move ourselves into the vascular plants. Notice once again, as we broke this up, we're starting with the non-vascular plants. That means they got to stay small. They're limited osmosis and diffusion. And then today we're going to talk about vascular plants, but seedless vascular plants. We'll limit our discussion today to just those. We'll talk about seed vascular plants later on. Some plants don't produce seeds. Right? Ferns, for example, they produce spores, but they don't produce seeds. Seeds come later on. All right, so let me, bryophytes, we talked about that. Non-tracheophytes, liverworts, because they look like liver. Hornworts, that's not a hornwort. There we go, mosses. Okay. So the last group in the non-vascular seedless plants are these little, neat little critters, the mosses. This is actually pictured. I don't think I took this picture, but this is a sphagnum moss, the one that I've talked about a couple of times. I see it out in the woods a lot. It's real pretty. It almost big beds of it. You almost think, oh, it'd be nice to kind of lay down on it, you know. This is the thing I was telling you that the native people used to use as anti-inflammatory because it would be wet, like a wet cloth, a wet compress. Now, remember some of the terminology we talked about the other day. We got the gametophyte. All right, the gametophyte is the haploid generation. It's that thing right there. And the gametophyte, if you pulled out one gametophyte, this down here in the lower right that I'm circling in the blue, that's a gametophyte. It's a small leaf-like. It doesn't really have leaves yet, right? We don't really get to leaves, but it does the same purpose around this stem. At the very end of that leaf-like structure, there are gametangia. Okay, so at the very tip of this leaf-like structure, up in here, and up in here, at the tips of the leaves, there are going to be gametangia. And the gametangia are going to be male or female. They're going to be an archegonia or antheridia, and they're either going to produce sperm or they're going to produce an egg. Okay? The sporophyte. Now, you don't often see the sporophytes, and I'm not real up on my moss when moss sporulation occurs, but next time you walk by a bed of moss, look at it a little bit more closely, and you might see, in addition to these green part, these little wispy brown things. I'll have to check and see if the mosses are forming sporophytes out in the nature trail. If I find some, there's that. So one that grows behind that huge sand live oak that always seems to have sporophytes. I have to check when I get back after next week and see if it's got spores. Take you guys out there and show them to you. It's really kind of cool. They're really small and wispy. Now, remember from our alternation of generation discussion, the sporophytes make spores, the gametophytes make gametes, but the sporophytes are the only diploid part. See this little wispy thing, and it's also nutritionally dependent upon the gametophyte. See, it's growing out of the top. It grows out. Let me erase some of my scribble here. It grows out of the top of this thing. Come on, pen. There we go. Grows out of the top. There's an egg. There's an egg at the bottom of the archegonia. The sperm fertilizes it, and the zygote grows out of the top. Now these sporophytes are going to eventually form a capsule. A CD is the thing that holds up a sporophyte. It's like that stick, if you will. And in that capsule, spores are going to be made. And when time is ready, that capsule is going to rupture. And those spores are going to be distributed out into the environment. And those spores are going to be produced by meiosis. So they're going to be variable. And then mitosis is going to kick in and start cranking out a lot of them. So we get the numbers. And guess what? The sporophyte growing tall. How do you think the height of the sporophyte, relatively speaking, we're not talking meters here, but what do you think the advantage that conveys to organisms is? You know, the taller they grow, the sporophytes grow. Heck yeah. Sure, they can spread out more, you know, they can disperse more of their seeds. And yeah, Melody, to be honest, sure, the stronger they were, the taller they could make those sporophytes, the more they could disperse, so they would reproduce more. Absolutely. Absolutely. Okay, sporophytes do work on their way up, though. In other words, they work before they become a sporophyte. This one here, what I'm circling, is green, so they can photosynthesize until they start making spores. And of course, like I said the other day, the reason why you're going to find these things in moist, wet environments is because of the fact that the sperm are flagellated. They have to swim through water droplets. Okay. Remember, these are not mosses, right? The one in the upper left is Spanish moss. It's not a moss. It's a flowering plant. And the one in the lower right is, it's not even a plant. It's actually a lichen. We talked about lichens, reindeer moss, which I think is kind of a funny name for stuff that grows in Florida. Also deer moss, even deer lichen. But don't put those in your field notebooks as mosses. They're not mosses. Okay, what I want to show you next is the specifics of reproduction in a moss. It's alternation of generation again. And again, when you hear that word, you should think, okay, what do you mean by that? Is alternation of two multicellular generations. One's haploid and one's diploid, but they're both multicellular. But I want to show you how this works in mosses, and I've also included some pictures from what it looks like under a microscope slide to kind of help you next week in lab. I mean, I'm not going to be there, but the pictures you see here that I'm going to show you, you might want to make sure you bring your notes, or if you have them on your laptop. If not, the computer there in the room, by all means, please just use that to search for these things. But this will help you kind of identify the structures you're supposed to look for. So let's start up here at the top with the male gametophyte. Okay, so we have a male gametophyte and we have a female gametophyte. Now that isn't the case in all plants out there. In some cases, a spore can produce gametophyte that actually has both male and female parts on it. That's how ferns do. Ferns have what we call ferns are homosporous. They produce one type of spore. It forms a bisexual gametophyte, not in the way you think, but bisexual in the fact that it has male and female parts on it. And then these mosses here are heterosporous, right? They make different spores. They make male spores. They make female spores, essentially. All right, and those form buds, and if they survive, they're not very big. Here's one right here, right? They're only one cell layer thick, so they're really using that diffusion osmosis to grow. But they grow into a male and a female, a gametophyte. And the male one has antheridia. And this is what the antheridia looks like under the microscope. So this is what I want you to use as a guide when you're in class. So the antheridia face up. The sperm are the darker purple little streaks that you see scattered throughout the antheridia. Those are tons of little sperm cells. And in the female, the archegonia, or the archegonium, which is singular, I don't know how well you can see in the picture. It looks like a vase. You're going to be looking at it from the side, and you'll see an egg. You can see an egg down there at the bottom sometime. So look for something, and sometimes the way they prepare the slide, the vase kind of gets chopped up a little bit, you know, but look for something that looks like a vase, and if you spot it very closely, you might be able to see a little dot inside that opening space at the bottom, which is the egg. Okay, so the sperm is going to be produced, and since it's flagellated, it's going to swim through little water droplets. Little sperm swimming from water droplet to water droplet until it finally finds its way to an archegonium. Down the vase it's going to swim, and when it does, that's what's going to happen, right? We're going to have fertilization, just like you talked about in animals. Going to form a zygote, a diploid offspring. And then that zygote, through mitosis, is going to grow. And it's going to get taller, it's going to get taller. It's going to remain nutritionally dependent on the gametophyte, though. See the brown part? That's the sporophyte. That's what the zygote is growing into. It's growing out of the top of the archegonium, but it's connected. And that's why we say that in mosses, the gametophyte generation is dominant. Again, that means nothing but behavior. It means it's the obvious one. And the sporophyte is nutritionally dependent. So it's a non-independent, or dumb way of saying it, it's dependent upon the gametophyte. Sporophytes grow out of the top of the archegonia. The CD is the little appendage or stick-like thing that holds them up. At the top, they have a capsule with sporangium, and a sporangium is covered by a peristome, which is the lid. When it pops open, we'll get spores, some up to 50 million spores. Take a look at this peristome down here. This is really cool. So this is a really high resolution, high magnification image of a spore, a sporangium peristome. So this is the cap part that would pop off, and the spores would, up to 50 million, would spray forward. Using your detective analysis here, what happened to my mouse? What do you think the little what do you think the little teeth are for? Why does it have all these little teeth arranged all around it? Keep in mind what I told you that it does. I thought this was kind of cool looking at this. If you're trying to think about like your garden hose at home. Yeah, it might be some protection. I don't know of things that feed upon spores or would damage it, maybe. What about that garden hose thing I said? Think about, imagine, especially if you have like a setting on your garden hose where you can adjust the spray. Yeah, right. As those spores are popping out of that peristome, remember some of them, like we saw it in the equisetum, have little springs on them. But as those spores are popping out, those teeth are basically spraying, causing it to disperse. All right, it's the same thing like if you watch it, water flowing by nice and smooth, and I put your hands in there, and that water becomes very turbulent, right? So that turbulence moves all those spores around. So I thought that was kind of cool. It's kind of like setting the spray nozzle on your garden hose to really tight, so it's a tight mist. It means you're really covering a wide area, right? Okay, now then after those spores find a suitable place to germinate, they first are a protonemata. That little thing is right there before they become a bud. Now buds grow these little hairs on them. They're not roots though. They're called rhizoids. Let me remind you that it's a technicality, I know, but you're not a root until you have a trachid. You're not a root until you're vascular. And we're still talking about non-vascular plants. By true definition, a root does two things. It anchors the plant and it takes up moisture and nutrients. Rhizoid only anchors. It does not have tracheids, so it's like a solid tube, if you will, if you think about it being a tube, kind of, versus one that has a channel in it. And again, I'm a horrible artist, but you get the idea. Okay. So bring that to lab next week. Bring this picture to lab. And again, I pulled these offline. You can find them on your computer, or by all means, please use the computer up front to do it too, okay? All right. So now we're done with the non-vascular plants. Let's talk about vascular plants. Here's Cooksonia. That's kind of cool, a fossil of Cooksonia. Remember I said soft-body things don't fossilize very well, so anytime we get a fossil of a plant, that's kind of a cool find. And down below is not actually a Cooksonia plant. It is a artist's rendition of what Cooksonia might have looked like. Cooksonia, again, didn't have any roots, but it was anchoring to the surface. So these organisms are going to be, those were the ones we saw in the original picture that were growing along the shoreline. All right, Cooksonia, remember, is the extinct one. Don't get that confused with the whisk ferns, which I'll talk about later on. But here's the sporangia. Okay, Aclophyton major is this critter's name, or was this critter's name. And so you can imagine that having things, rhizoids, only for anchoring, in other words, not being able to absorb anything through these little structures, that you were probably limited to moisture-type habitats, right? So, as it says here, the bryophytes were doing really well and still are. I mean, they were around for 100 million years, but the non-vascular, or the vascular plants started appearing about 425 million years ago. And again, based on what we were talking about at the beginning of the lecture, there's a lot to do with how the habitats were changing. The habitats were changing, and the diversity of them was changing. We're also going to lose the dependency of the sporophyte. We're actually going to see this gradual trend in plants from, or not trend, I guess, but this difference in evolutionary trend where the gametophyte becomes less dominant and the sporophyte becomes more dominant. Now, if you're thinking, why would that be? The gametophyte, remember, is haploid. And you can be haploid. All a cell needs to have to work is genetic information. You don't have to have two sets of chromosomes. Remember, being diploid, like we talked about in evolution, kind of gives you a backup plan, maybe, that maybe works, right? Having that like heterozygote advantage and those things that we all talked about back in evolution. But if you're okay and you can eke it out as a haploid, and that genetic information you have works fine, well, by all means. Now you're going to be kind of limited, right? You're going to be kind of limited how you can grow. The world is a harsh place. So as these organisms, as these plants move into harsher and harsher habitats, you would think, you might surmise, that it makes more sense to put more effort into the sporophyte, to start letting the sporophyte be dominant because it's diploid. So it has a backup plan. So it has the ability to tap into those recessive alleles that might be beneficial in a particular habitat, right? All that stuff that we talked about. Still, you're not getting very tall, though. You're vascular, okay, but you're not going to be getting very tall. You're still saying only a few centimeters tall. No real roots, no leaves. You're photosynthesizing through this tube part. Okay, there are stomata. This is actually a fossilized picture of a fossilized stomata. Remember stomata, carbon dioxide and oxygen. So we're beginning to see more and more of this. There were stomata in some of the bryophytes too, but we're beginning to see stomata. What's really cool about this too in the fossil record, when you trace stomata in the fossil record, when we see that a lot more plants start having a lot more stomata, so in other words, the number of stomata increases on plants, it gives us evidence that the carbon dioxide levels on Earth were changing. So we'll talk about that a little bit later on, but it's cool. It's kind of that detective stuff. Okay, so everybody good with Cooksonia? What I want to move into now is kind of jump from, you know, this vascular plant into vascular plants that exist today, so extant vascular plants. So what are some of the traits that came about through time, you know, some of the changes that were occurring, some of the structures that formed that, you know, that really proved to be successful, at least in these groups of vascular plants. All right, so we'll break it down to 5 things. We'll talk about sporophyte dominance and independence. We'll discuss something called xylem and phloem. Then finally true roots and leaves, and then what we're doing to the spores, how we're getting this spore variation and how that's going to help plants out. So let's start with this first one if there aren't any questions first. Let's go on the sporophyte dominance, right? Questions? Hi, Dustin. How are you today? Good. All right, so as I said, if you're walking through the woods and you look down at a moss, you see this green bed. What you're looking at is haploid, a multicellular haploid structure. Each one of the cells that makes up that green moss is haploid, but it's multicellular. It's the gametophyte, okay? And the sporophytes, as I said, are these wispy little things. They're the only diploid part, and they're only around long enough to make spores. So that's what I mean about sporulation, whenever that occurs. So gametophytes, spores arise out of gametophytes. They make spores, and new spores try to make new gametophytes somewhere else. Where we've moved to from there now is with the group of plants, the vascular plants, or the tracheids. Trachid and vascular means the same thing. Think about that. Think about the official name or the technical name for your windpipe. What is that? What is the technical name for your windpipe? Yep. A trachid is a tube. Okay, so notice what happens as we move. And this is a fern, for example. The frond of the fern, remember, it's not really called a leaf, but the frond of the fern is the sporophyte. Remember the picture I showed you on the back of it? You saw those dots? That's where spores are produced. So if you're the thing that holds the thing that makes spores, right? If the sporangia are found on you, then you're a sporophyte. Okay? The gametophyte, unlike in the mosses where it's very prominent, is very small. This is a picture of a gametophyte. Now, this is nowhere near scale, folks. These things are really small, really delicate. I've dug around, you know, my ferns. I've never seen a gametophyte. They're real easy to destroy. Now, you'll look at something like this under the microscope. It's not going to look exactly like this, but it'll have this heart-like structure. Right, that's the gametophyte, and actually this is a bisexual gametophyte, the male and the female structures, and I'll show you later how to help identify them. But this is the sporophyte growing out of it. This is going to grow into one of those leaves. Not leaves, excuse me, fronds. You notice how much more, you know, what the difference is in terms of which generation is dominant now, or prominent maybe is a better word, right? The sporophyte becomes much more prominent. Because again, you're dealing with tissue that has a backup, right? It's got alleles. And again, it does not mean that one's better than the other, right? Or one was a replacement for a defective system, because again, there are lots of mosses around today, okay? I always want you to keep in mind that evolution does not have a goal, that there are many organisms that are still using those less sophisticated ways and doing quite well with them. Tracheids, vascular plants, yes, it's about the tracheids. It's about the xylem and the phloem. Now the way I remember these in college was xylem up, phloem down. Xylem up, phloem down. And xylem with the X and the Y kind of looks like it's pointing up, or has the cup shaped up, if you will. The xylem in the picture just to your right are going to be these cells. And the phloem are going to be the outside, the darker colored cells. And in the tree, the xylem, again, is the more interior part, and the phloem is the exterior part. And the xylem, as I said, the xylem up, so this is a cross section. right? Like you're looking through a stem. Here's xylem there and phloem on the outside. The xylem is going to carry water, going to carry minerals from the roots, right? From the true vascularized roots up through the xylem up into the rest of the plant. So now you're not limited to just diffusion osmosis. You can get bigger. And you can also reinforce the walls of those cells, of those tubes. You can lignify them. Remember I told you about lignin? Lignin is one of the most durable substances on earth, man. It's like concrete. So you can not only stiffen each one of those individual cells, take a bunch of those individual cells that are stiffened together, and you got yourself a tree, don't you? And the phloem brings the stuff back down, like the sugars and the proteins and all the products that are made. So maybe not phloem down, but more like phloem throughout. When they tap the trees for sap to make syrup, they do it in the fall, because that's when the tree is getting ready to lose its leaves. This is all growing up north, and the sap is going to be carried down into the lower parts of the tree and the roots to be stored. Well, if you come along and you cut into the phloem, you catch that sap running down the tree, and then you drain it off, and that's how they make syrup, right? I saw them do that up in Vermont. It's actually kind of cool. My wife says, "You want to go see on a tour of how they make syrup?" I'm like, "Really?" But it would, they showed out on the trees too. I thought that was kind of neat. And we got fruit pancakes. Okay, so everybody understand the implications of the xylem and the phloem, right? So allowing the lignified in these tubes means you could grow taller, you could grow bigger, which means you could outcompete each other. And as you got taller, like with the sporophytes, you could spread your offspring further away. Right? All things that would have contributed to a higher rate of success, right? Okay, so now we're going up with the xylem and the phloem up into the root shoot part. Let's go down into the roots again. Now, roots again, and I put it in red to remind you, have two purposes. A true root must anchor and allow for nutrient and water absorption. Okay? Now, roots may have evolved from subterranean stems, things called rhizomes. Rhizomes are sometimes called horizontal roots, but they're not really roots. Like in this picture, this is like, could be crabgrass or any grass. It's St. Augustine, actually. Where's that picture? Now look at that. Here's St. Augustine. See that tube-like thing that's running? It's sort of purplish in color right there. That's the rhizome. That's the runner. We call it a runner, huh? I got to go all the way back to. Can I go back to the slide I was on? Right. So you've seen that in your St. Augustine grass, right? If you have St. Augustine, everybody's seen that. So that's a way. That's what we call vegetative reproduction, because they're not. It's not a matter of gametes getting together, but they're reproducing by just growing long. So maybe that's where, that could be where the roots evolved out of. And you can see, like in these rhizomes, the roots are coming out of the bottom. Like I said, rhizomes are usually considered horizontal roots, although they're not roots. They do some anchoring, so they're more rhizoid-like, and roots grow vertically. We think that they may have, roots may have evolved from rhizomes, but it would be, if it did happen that way, it would be because of convergent evolution. Let me go forward a couple of slides and I'll show you what I mean by that. If we go to here, roots were already in lycophytes when they started evolving. Lycophytes, they're already in lycophytes when the ferns and the other plants started evolving them. So roots evolved, in other words, weren't here and ended up showing up in both of these. See what I mean? So convergent evolution, right? Okay. And there's a fern plant, and you can see there's the rhizome part, and then these are the roots. So don't confuse rhizome with rhizoid. A rhizoid are the little anchoring things that can't absorb anything. The rhizomes are those horizontal runners. Rhizomes are runners. Okay, now, about 360 million years ago, the atmospheric carbon dioxide levels begin to drop dramatically. And the reason why that was is this is right in the middle of the Carboniferous, all right? This is right in the time when, man, the plants are going crazy. As I said, this is the time when, we see that really thick layer of carbon down in the rock layer. We know there were a lot of plants growing back then. So, and we can detect carbon dioxide levels. And so we know that the carbon dioxide levels are dropping. So natural selection started to favor larger leaves with more stomata. See, I told you more stomata earlier can be used to tell about levels of carbon dioxide. Lower carbon dioxide levels, how would you respond? Make more stomata. So you can bring in more carbon dioxide, right? Okay, so let's look at the leaves part first. When you make your leaves bigger, you increase your area for photosynthesis. That's going to be good, obviously. And of course, you can imagine this being competitive among plants, as one made a bigger leaf than the one below it, then this one wants to have a bigger leaf and a bigger leaf, and so there's an arms race right there. Well, the plants known as the lycophytes, Krause's spike moss, that's a lycophyte. Those organisms, what they did is they made small little microfills. And again, they're still around. Krause's spike moss, that picture you see there, that's an organism that exists today. But each of these little V-shaped things, those are the leaves. They're single branched, one piece of vascular material moving off into the micropyle, right? And that works. They photosynthesize. You can see down below how we think it came about. It was basically kind of an off-budding that occurred. Some genetic deformity, let's say, advantageous, obviously. And then you can expand out that leaf and you can increase your photosynthesis. Again, you're not going to get real big, though. So those are the microphiles. Now, only the lycophytes have those. The megaphiles is what we find in all other plants, including the plants that we're not going to talk about today. So the big trees and all that. Down below is how we believe megaphiles arose. So we get a branching stem, same idea, right? So maybe that branch that occurred in the microphiles happens more than once. And first it's kind of branching like a bush. And then eventually through time it begins to branch so it's in the same plane. You see my fingers? I'm trying to show you that all the branches are in the same plane. And then once that happens, tell you what, it's real easy to put tissue between those, isn't it? It's real easy to start filling in between. Look at that. Real easy to start filling in between there, making bigger leaves, more photosynthesis. Right? And those are megaphils. Look at this. Look at this Tunbridge filmy fern. If for no other reason, that's a really cool name. Look at that leaf. Isn't that cool? I mean, you can see it right there. You can see the little vascularization and all the tissue in between. So cool. So neat, right? Okay. However, here's the thing. For every innovation, for every evolutionary adaptation that occurs, there's a cost, right? There's a price. And you know, it happens in life all the time. For every 2 steps forward, or maybe even 3 steps forward, there's going to be 1 or half a step back. So here's the problem. Larger leaves certainly helped with greater photosynthesis, okay? No doubt. That's great. But larger leaves absorb more heat, okay? And this would have been really, really a problem in plants in the tropical region, okay? Or in the region where the sun hits the earth more directly. Now, without factoring in the tilt of the earth, because it makes it a little too complicated. Remember, and when the sun strikes the surface of the earth, at near the equator, it hits the surface of the earth in a circle. But because it's hitting the higher and the lower latitudes at a glancing angle, it doesn't make a circle, it makes an ellipse. And then ellipse has a greater area. Same amount of sunlight, but in a greater area. That means there's going to be less density of sunlight. So plants living near the equator, you know, growing big leaves, man, because you had that extra sunlight, that's fantastic. But here's a problem. There's a lot more sun, which brings in the radiant energy you need for photosynthesis, but it also brings in heat. And just like you and me, plants have to worry about overheating. They have enzymes too. Remember all those enzymes in respiration and all those enzymes in photosynthesis? All enzymes can be affected by heat, right? They can be denatured. So how do you're helping yourself in one way, but hurting yourself in the other way because bigger leaves get too warm. So what can you do? Well, if you remember back in Bio 1, those stomata, they open and close to exchange carbon dioxide and oxygen, but they can also lose water. It was the reason why there were C4 in CAM plants, remember? Because sometimes losing too much water for your stomata can be a problem. But if you lose not too much, let's say, if you lose, less than a critical amount, you can actually use the physics when water evaporates off the surface. I don't care if it's your skin, a leaf, or the sidewalk, folks. Water removes heat from that surface. So evapotranspiration. Evapotranspiration is plants sweating, if you will, kind of helping themselves cool off. Okay, so if you have larger leaves, you have more stomata, you can bring more carbon dioxide, and you have more stomata to release more liquid to help cool off when it's getting warm. All things that you might expect would be advantageous variations when you're moving into harsher climates, right? Think about what larger leaves would help you if you were moving into a climate where it was great for rain, right? Lots of rain, a lot of moisture, but a lot of clouds. Well, a lot of clouds, less photosynthesis, make bigger leaves, make a bigger receiver. It all fits. That's what I love about telling the stories, because it all is so cool how it fits. Like those penguins walking 70 miles over the ice, a little bit at a time, little tiny changes adding up to millions of years, right? So we got the leaves, we got the roots. How about this? How about what we started doing to spores? Okay, now, a sporophyll, phyll means leaf. A sporophyll is a leaf that produces spores, okay? Now, in the mosses and the liverworts, we didn't have leaves. We had a thallus, but it wasn't really a leaf. But in essence, for the plants that we're going to talk about, a spore-producing structure is a modified leaf. As a matter of fact, when we get into the cone-bearing plants, like you see the pine cone there, and the flower-bearing plants a little bit in our next chapter, flowers and pine cones, those are just modified leaves. So Mother Nature, plants do that a lot. They modify their leaves. Like, for example, in a cactus, I think I told you guys when I took you out on the nature trail, in a cactus, the leaf is the spine. The part that photosynthesizes, the big green part, that's actually the stem, right? The stem of the leaf. So in this case, cactus said, hey, I'll tell you what, I'm going to take my leaves and turn them into shark things and keep things away from me. And then I'm going to just expand this stem of mine and make it photosynthetic. Because stems are green, they photosynthesize, right? So any leaf that bears spores, whether it looks like a leaf or it looks like a pine cone, like you see in that picture, is called a sporophyll. Okay, and on sporophyll there are sporangia, and in ferns those are those dots again, right? Sorus or sori. Okay, just like you saw, there they are. So the sporophyll or the sporophyte, all right, the sporophyte, the sporophyll is the leaf that contains the sporangia. In lycophytes and in a group of that we call the gymnosperms, the leaf has been modified into a cone. So this is a lycophyte down here, and they make these things called strobili, or strobilus is singular. And I've never actually seen a lycophyte up close, but the reproductive part that you see on the strobili is a cone shape. And maybe more familiar with these guys. These are the pine cones, right? And since strobilus doesn't really, kind of picture in your head, it's Latin for cone. Strobilus is Latin for cone. Look at those pine cones that I just circled up there. Those are off the same tree. What's different about those pine cones? Yep. Or the pine cone contains sporangia. The pine cone is the sporophyll. That's the modified leaf. Yep. So take a look at those pine cones up above there in that pine tree. Tell me, those are both pine cones. Why do they look so different? Same tree, same species, same age. That's right, Harrison. Which one's the male? Excellent. Which one's the male? Think about it in terms of us, Melanie. Think about it in terms of humans. Which is bigger, an egg cell or a sperm cell? Right. So which one now do you think is the male cone? The smaller one, right? That's the pollen cone. Pollen is sperm, folks. That's what the pollen is the plant version of sperm. It's the same thing. So this is the pollen cone, and this is what we call, and we'll learn this again later, so don't worry about it. It's called an ovulate cone. So this is where the egg is going to be, and this is where the sperm is going to be produced. Okay? Now, seedless plants. Let's get some terminology down. I made this little chart down below to try to help you with this sometimes overwhelming and kind of confusing terminology. So seedless plants, right? So back to mosses for a second. Sporangium are on sporophylls, all right? So sporophylls are the leaf-like structure. Now, the seedless plants don't have leaves, but the leaf-like structure that produces the spores, right? So, and they have a single type of spore. The sporangium produce a single type. They're homosporous. And those homosporous spores, when they germinate, whoo, that was a mouthful. When they germinate, they form a bisexual gametophyte. In other words, one structure that has both the male and the female parts on it. And I'll show you that when I get to it, okay? And a bisexual gametophyte, that one structure both produces eggs and sperm. Now, in seed plants, okay? So now we're talking about pine trees, flowering plants. Most of the plants you're familiar with, they have what we call heterospores. Heterosporous. Heterospores means there's two different types of spores. So, in keeping with the size thing, I tried to highlight in red. The megasporangium on a megasporophyll. So again, if we were wondering where in these pine trees up above, where's the megasporophyll? That's the megasporophyll. Megasporangium on that cone, on the megasporophyll. Megasporangium are made up of megaspores. Megaspores are made up of female gametophytes. See, the gametophyte in these is very, very small. It's actually contained microscopically inside that megasporophyll. Those produce the eggs, right? Mega, mega, mega. Female, big, right? Microsporangium on microsporophylls make microspores, which make the male gametophyte. All right, and remember, don't get all confused here. Spores are still making gametophytes. Whether you're talking about homosporous or heterosporous, and gametophytes are still making gametes. We're still dealing with spores. We haven't removed anything about the alternation of generation. Plants all the way up until the ones at the very end, the flowering plants we talk about, all have alternation of generation. Right? They have this gametophyte and this sporophyte generation. Okay. Ready to look at some of these vascular plants now. Any questions on those innovations that came about? So let's here we are, right here. We're going to look at lycophytes. What time is it? Oh, good. Okay, lycophytes, the club mosses, spike mosses, which I put in quotes because again, I want to be correct, and I want to remember that you guys don't forget that a true moss is a non-vascular, seedless plant. We're still seedless here, but we're not non-vascular. The quillworts, they're down below. Right? There's a spike moss. There's a quillwort. There's a club moss on the right-hand side. Looks like a club moss, right? These are the sporophylls. So these are the leaves that contain the sporangium. They're going to make the spores. Some of these organisms, there's not a whole lot of them. Some of them that are around today are epiphytes. What's it mean when a plant is an epiphyte? What does an epiphyte do? Give you a hint. Orchids are epiphytes. Ball moss is an epiphyte. Oh, Spanish moss is an epiphyte. No. How does Spanish moss grow in terms of? It grows on what? On the ground with roots? Yeah, it grows on other plants. That's what an epiphyte is. Okay? Orchids are the same way. Orchids grow on other plants. Doesn't mean they're necessarily parasitic. Spanish moss is not parasitic. It just grows on other plants. And they're called mosses because I guess, they are kind of moss-like, if you will. This is what it looked like during the Carboniferous, by the way. 359 million years ago. Look at these horsetails. Now, there are horsetails around today, but they're small little plants. These things were huge, massive ferns. And lycophytes today, which are small little plants that kind of stay low to the ground, back then they were these trees. And of course, my favorite of all, look who's around. See a little insect there? Dragonflies. Dragonflies are pretty much the same now as they were back then. I love dragonflies. Every time I see a dragonfly today, I think, wow, you guys, your lineage witnessed the dinosaurs, even before the dinosaurs, right? Now, club mosses are homosporous, spike mosses, quillworts, they're heterosporous. So we're seeing, these other organisms, experimenting with different types of sporulation, different types of spores that they can produce. All right, any questions on those guys? Small little group, the lycophytes. Lycophytes with their small little lycophils, with their little microphils, unlike we're going to find in some of the other ones. The manilophytes. See, here we are. The manilophytes are the ferns, one of my favorite. I have a lot of favorite plants, but the horsetails, and then the whisk ferns. The whisk ferns are cool. I'll show you the whisk ferns. Those are the ones I've been talking about that are a living fossil. Well, there they are right there. The ferns, the horsetails, the whisk ferns, this is the closest group to the seed plants versus the lycophytes. See, what we mean by that is manilophytes and seed plants. There's the seed plants. They share a common ancestor 375 million years ago, whereas ferns share a common ancestor with lycophytes somewhere around 425 million years ago. So ferns are more closely related to seed plants, the rest of the plants in the other chapter, than the lycophytes are. The lycophytes, of course, include the ferns, a very common one. The horsetails, even though they're giant, they're not really giant. Tried to upload my picture of my horsetails, but it didn't work. I was coming back from wandering around the woods the other day and happened to pass a place where they a nursery where they grow palm trees for folks, and I looked in the ditch and there were all these horsetails. It was incredible. These are the little leaves on horsetails, and horsetails actually make two stems. They make a vegetative stem, the green one that photosynthesize, and then the strobilus, that's the sporangium on these guys. That's where the spores are made. You see that little whisk fern there? I got a better picture of it a little bit later on, but that's the thing that looked like Cooksonia. Remember I was telling you that's a living fossil? It's not related, right? But it looks a lot like it. It's like I said, it's convergent evolution over million, it's separated by millions of years. Very, very, very cool. Very cool. Okay, ferns, they're the most widespread. 12,000 species of ferns. Some get very tall, these tree ferns. Most are homosporous, so most make a bisexual gametophyte. That's the gametophyte you're going to be looking at under the microscope. And I have some pictures I can show you of that too. Tons of tons of spores, over 1 trillion spores in a lifetime produced by your average fern. The sporophyte is conspicuous, right? It's the dominant. The gametophyte is that little tiny heart-shaped thing that I showed you that you look at under the microscope. Both are green, both are photosynthetic. The sporophyte or the fronds are called that because that's where the sori are, right? The little dots that are on the back of the leaf. But I want to make you aware that there are exceptions. This is a cinnamon fern. Now, cinnamon ferns don't make those little dots on the back of their leaves. Cinnamon ferns produce their spores or their sori, okay? They produce those on a separate stalk, a separate structure, a separate frond. It's called a reproductive frond. This is the reproductive frond. The reason why cinnamon ferns are called that is because that reproductive frond, and this is a picture of what I took across the street. I'll have to check out the nature trail to see if the cinnamon ferns are producing reproductive fronds. It looks like a piece of cinnamon. It has the same color. And actually, if we do find some, if we can get some, you can look at them under a microscope, and under the microscope, that cinnamon-looking thing, it looks like sori, again, just kind of all curled up. They're just not found on the back of the leaf. So not everybody does it the same way. Okay? All right. Now, let me show you the alternation of generation in a fern. So this is what you're going to be looking at in a microscope, and then I've got some pictures to follow up with on this. Let's start with the gametophyte. Here it is. Here it is, the mature bisexual gametophyte, okay? Bisexual again because it's got antheridia and archegonia. Now remember this when you're looking under the microscope. You're going to be looking at one of these things like this. The Antheridia are going to be near the end, opposite the little dent. These little things these are rhizoids, not roots. These are for anchoring. Ferns have roots, right? This is a root. Horizontal roots too, so they do have runners, but the little rhizoids are just for anchoring. The Antheridia are going to be up near that end, and the Archegonia are going to be up near closer to the dent. All right, so that'll help you identify them. Sperm are produced in the Antheridia. They're flagellated. They swim to the Archegonia, where out of the side, very much like we saw in the moss, a zygote grows. And out of that zygote grows, or that zygote develops into a sporophyte. And that sporophyte initially, as it begins to develop, will look something like kind of like a clover, and then it looks like a fiddle head, like the top of a violin. See, and you can tell what it's doing, right? It's unrolling. So this thing is going to unroll, and then as it unrolls, the side leaves are going to pop out, and you get your fern frond. And on the back of that, if it's that particular species, that's where the spores are going to be again, right? There's the sorus. That's one. Sori for many. Sporangium. Each sorus is made up of several sporangium that are making spores through meiosis. And we're back to the beginning again. Spore dispersal. Spore dispersal. Remember I told you about spore? So you can spray them out. The taller the sporophyte gets, the taller the fern gets, the more dispersal. Or like in the the horsetails, they did it with a spring, right? Remember that elater thing? Now go back to the bisexual gamete, or I'm sorry, the bisexual gametophyte for just a second, right? So the spore germinates into a young gametophyte, and eventually we have a bisexual gametophyte. You would think that having both sex organs on the same, in such close proximity, right? This is kind of what it looks like from the side, by the way. There's the rhizoids. And there's the root coming out of the sporophyte. You would think that that would increase the chances of inbreeding. Well, nature's pretty clever. What she does is the Antheridia, the sperm mature first before the eggs. Okay, so in the Antheridia, the sperm are produced, and then they're dispersed out into the environment. So they have a time to kind of get away from their own gametophyte to find a gametophyte further away that is presumably less related or not related to them. Now, does that stop all inbreeding? No, of course not, but it does prevent some of it. So there are some ways of getting around this. Okay, so this is another good picture or set of pictures you might want to keep for lab next week. There's the gametophyte. That's not a really good picture of them because it actually looks more like a heart-shaped structure. But the Antheridia, the male part, always look like the soccer balls to me. Right? They look like soccer balls. This is where the sperm are going to be produced. Now remember, those are going to be down near the rhizoid. They're going to be away from the dent. They're going to be down on this end. So this is going to be the male part. They produce flagellated sperm. Flagellated sperm are going to make their way to an archegonium. Now the archegonium are going to be up near the V a little bit further, and they're going to look like, in the microscope appearance, you're not going to be looking at a vase from the side now, like you were in the mosses. You're looking straight down the vase. See, there's the egg at the bottom. So you're looking down through the top of the vase. That's the archegonia. Sperm swims into the archegonia, and if it fertilizes one of those eggs, this grows out. That forms a fiddlehead, and that unrolls into a new frond. And then that frond produces sori. And those sori have sporangia, and the little purple things are spores. Now the spores germinate, and we complete the cycle. Okay, so again, this might, and you can always, as I said before, go on up to the front and just, the guy's going to be proctoring the class. Scott's a really bright guy. Now, how much he knows about a fern or moss morphology, I'm not really sure, or structures. But by all means, just go on up to the front, type it into the computer up there, and put it on the screen, and everybody can take a look at it. Type in, fern Antheridia. Type in what your slide says, and it'll help you kind of orient yourselves. Okay. Ooh, the horsetails. The horsetails grew really large in the Carboniferous, but today it's kind of a specialized plant. There's not many of them around. There are actually only about 15 species, and they're all pretty closely related. They're all in the same genus. They're all Equisetum. Horsetails, equisedum, equus is horse. The reason why they're called horsetails is, I guess, because a tail of a horse, if you've ever felt it through the hair, is very stiff. Okay, and horsetails are kind of stiff. They're homosporous, so they make a bisexual gametophyte, and the stiffness is because impregnated in the tissue is silica, a granular glass-like substance. It's gonna deter things from eating it, right? It's kind of like chewing up something that's been dropped into sand. If it doesn't completely deter you from eating it, you're not going to be able to eat a lot of it, so you're going to grind your teeth down pretty doggone quick. A rush is a type of plant. It's actually a type of grass, although horsetails aren't really grass. But a rush is a round type of grass. And so sometimes horsetails have been called scouring rushes. So they're not a grass, but they're round like a grass that is a rush. And the scouring part is because you could actually, and pioneers did, they would take them and use them to scrub their pots out. That was the first thing I did when I found those horsetails alongside of the road. I went up and I felt them. And you could feel it. It's gritty. It's really kind of cool. Okay, so there's the fertile stem again with the strobilus and the vegetative stem, and then the little tiny leaves that are sticking out. That's what those little things are. The whisk ferns. This is that living fossil again, right? This looks like Cooksonia. And notice, interestingly enough, it's found in the subtropics and the tropics, which we think that's where Cooksonia probably evolved. Right, so again, it's a living fossil, but it's not very related to Cooksonia. It's actually related to ferns. It has no true roots, just like Cooksonia, but we think that it did at one time. It probably lost them during evolution. Now, if you're wondering why a plant would lose its roots, again, it's because of the environment. In the tropics, we normally think about the tropics as being very productive, right? A lot of stuff grows there. But we know that there's this reoccurring problem in the tropics where farmers cut down fields, cut down tropical rainforest and try to turn it into agricultural fields. And within a couple of years, the dirt is no longer useful. There's no nutrients in it anymore. Really, in a tropical environment, because the rate of decomposition is so high, there's so many decomposers, it's so wet, there's a lot of fungus, a lot of bacteria, most of the organic matter, most of the nutrients is actually in the living material. It's actually in the plants. There's not a lot in the soil. So subtropical soil, more tropical soils, even Florida soils, we're subtropical, they're not that dark, rich soil like from up north. So having roots for the absorption of nutrients, if you're living in a tropical environment, may not be necessary. You still need the anchoring part, right? So you're going to keep the rhizoid thing, but you're going to lose the roots because you're going to do most of your moisture absorption probably from the surrounding atmosphere. It's very moist in the tropics, right? Humidity. And your nutrients, once again, are going to be absorbed in the same way, right? So you're not going to rely on roots. And Mother Nature's got this thing with evolution that, sure, innovations come along, but sometimes Mother Nature does a little culling. She likes to simplify things down. So if you don't need roots, then don't make roots. Okay, so that's kind of what the whisk fern has done. But I think it's really kind of cool if you think about the living fossil part. These look a lot like, almost exactly like Cooksonia, but only because it shows how much similar environments shape things. Okay, so the last slide here, let's kind of finish up with what the Carboniferous was doing, right? So during the Carboniferous period, we get this massive growth of plants, like you see up above there, kind of like it looks like a cypress swamp today, a little bit more primitive. So we get this massive growth of plants, extensive CO2 level drop, right? Temperatures drop. Ooh, okay, so it starts to cool off, right? A global climate change, or specifically the warming of the atmosphere and the seas and everything, is caused by carbon dioxide levels rising, a lot of that being anthropogenic, human-caused. Okay, but you can see in a little chart there, just about the end of the Carboniferous period, the carbon dioxide levels drop dramatically. Okay, and when they drop dramatically, the temperature drops dramatically. All right, there was also the fact that during this time, a lot of the atmospheric carbon that was being incorporated into geological material was washing into the sea, and that was being incorporated into the calcium carbonate that made up the shells of mollusks. So those two things were contriving together to lower the carbon dioxide levels, and the temperatures on Earth started to drop. Now that means the Earth is going to start changing. Okay? One of the things that's going to happen at the end of the Carboniferous as the temperatures on Earth start to drop is the Earth begin to dry up. Okay? And when it starts to dry up, habitats become more harsh, more difficult. So that's where we're going to move into in the next chapter. That's where the seed plants increase in dominance. Seed plants develop their own set of innovations to deal with these harsh climates that we're forming. Remember I told you at the beginning the Carboniferous was called that because of the plant growth. Coal, 6 million tons of coal a year are burned. All right, 6 million tons. Now just to remind you, this is how coal is formed. Organisms millions of years ago, dying and decomposing, form peat, and through time, heat, and pressure, that peat gets compacted into lignite and then eventually into coal. So when we are burning coal, all that carbon dioxide that was incorporated into those plants is being released back into the atmosphere. And as I said, carbon dioxide in the atmosphere causes temperatures to rise. Okay? All right, well, that's good timing. That's it for chapter 29. Chapter 30, we'll look at the seed plants. Again, remember, next week, your exam on Tuesday is 27 and 28, prokaryotes and protists. And then your lab is the first part. You won't be doing the whole lab, folks. You'll only be doing the lab up to where it says gymnosperms. Don't do past gymnosperms. You'll see it in your lab manual, okay? So I'm going to stop the recording.